Okay, for this video, I'll be going through LSS Sec 1, separation techniques, the worksheet, okay? Okay, so the mixtures can be separated by physical methods, right? Because mixtures are not chemically combined, right? So we can actually separate them using separation techniques based on their physical properties. So basically, all these separation techniques are physical separation, okay? So the first one is magnetic attraction. So basically is to separate magnetic materials from non-magnetic materials. Okay, so we have many non-magnetic materials, so maybe it's better that we memorize what are the magnetic materials. So we have iron, okay, cobalt, nickel, and we have steel, right? So I've written down the chemical formula, okay, chemical symbol for iron, cobalt, and nickel. But why is it that I didn't write for steel? Because steel is an alloy. Okay, it is made out of either iron plus carbon or some other things, right? So they are not in fixed proportion, okay, and this alloy is also a mixture. Okay, however, it has the property of being a magnetic material because it contains iron, right? So examples of mixture separated by magnetic attraction, your iron filings from sand. So if you see any of these materials being mentioned, you know that it is talking about magnetic material, so you must use magnetic attraction to separate. Okay, next is filtration, is to separate an insoluble solid from a insoluble solid from a liquid. So we can say that this insoluble solid is the solute, okay, and this liquid will then be the solvent. Okay, however, in this case, the solute, it is insoluble in the solvent, so we can use filtration to separate it. So examples will be your chalk and water and sand and water. Okay, you have done this experiment in class, in the lab. Now, as the mixture is poured through the filter paper, the liquid filtrate. So the thing that comes out, okay, the liquid, it is known as the filtrate. This is important, all right? And what is left behind is an insoluble solid, which we call residue. Okay, so as you can see, the larger particles are not able to pass through the holes, right, in the filter paper, so they will be left behind. So these are the residue. But the liquid, being smaller particles, they can actually flow out, the solvent, the soluble particles. Okay, so how does this work? Because the filter paper has tiny pores, which allows the smaller liquid particles to pass through while trapping the larger solid particles. Okay, so you might need to learn how to draw, okay, because we have done the apparatus drawing in chapter 1. So basically, this is my filter funnel. Of course, you draw a straight line, then we have a V shape to show the filter paper. Okay, and usually we can stand it on a conical flask, okay, so that the smaller liquid particles can flow through, and which we call it the filtrate. What is left behind the filter paper is called the residue. So what are some applications of filtrations or this? Okay, now next is evaporation to dryness. It is used to collect the soluble solid left behind. Okay, soluble solid left behind. What does this mean? It means the solid has dissolved. Okay, but now we want to get back the solid. Okay, so we can't use filter paper because for the filtration, right, while using filter paper, the solid is the one that doesn't dissolve. But now, how about those solid that is able to dissolve in the solvent? We have to use evaporation to dryness. Okay, so you again need to know how to draw, at least for your wire gauze, okay, and your tripod stand. Okay, please don't use arrows like me, right? You should be using straight lines, okay, to point to the object. Then we have this evaporating dish. This one is the evaporating dish, and we have a water bath. Okay, so I'll explain the water bath first later. So solid left behind after heating will be the residue. Okay, so this is the one that we want. So examples will be salt from salt solution. Okay, now what is a water bath? So as you can see, the bubbles are forming, which means the water is already boiling. Okay, sometimes if we don't have the water bath and we put the solution directly to the heat, right? Example, directly to the heat. Okay, the heat may cause the uh, solute to decompose. That means to break down. So one good example is your sugar solution. Okay, as you know, sugar dissolves in water. So while the water has evaporated, now that if I don't use a water bath, the water evaporates, right? The water evaporates, leaving behind the sugar. And as you know, sugar, if you expose it to direct heat at too high a temperature, it will actually become charred. It will become carbon. So that means we cannot get back the sugar crystals as a whole. Okay, so therefore we will need a water bath. So this is a common question. Why do you need a water bath? Is to prevent the solute from being to undergo thermal decomposition. Okay. Next is your simple distillation. Simple distillation is used to collect pure solvent from a solution. Okay, that means we want the solvent. What is solvent? Solvent is something that we use to dissolve something. So examples of mixture separated by simple distillation is your pure water from seawater. So how do we get, what are the things inside seawater? So if we put it very easily, we have salt and water, right? Because your seawater is salty, right? Okay, so if I want the salt from the solution, right, or the mixture, I want the salt, I will need to use evaporation to dryness. Okay, evaporation to dryness. Meaning to say, I let the water evaporate, leaving behind the residue of salt. Okay, but now if I want the water instead, I don't care about the salt, okay, or whatever it is inside the seawater. Okay, I want the water instead. Okay, then I will have to use simple distillation. Okay, so let's look at the setup. We have a distilling flask. You need to be able to label the parts. I doubt that they will ask you to draw at Sec 1 level. Okay, so you need to know how to label the parts. So we actually have a thermometer, we have a distilling flask, we have boiling chips, we have condenser. All of them have a certain function. So you need to know the name as well as the function. So for A, the solution in the distilling flask is heated until it boils. Now the hot vapor form is passed through a condenser. So this hot vapor will come out, okay? And what is the boiling point of water? It is 100 degrees. So if I see that the thermometer has reached 100 degrees, okay, that means I know the water vapor is forming. Okay, anything below that or higher than that, it may not be water. So I'll wait for the temperature or the thermometer to reach 100 degrees. Okay, so once I do that, I can start my collection. Okay, collection of what? The hot vapor will be passed through the condenser. As the name suggests, it is condensing the hot vapor into from gas into liquid. Okay, so that it will drip out from here. Right, so this is the purpose of the condenser. The hot vapor cools and condenses into a liquid. Now this is important. It is not called a filtrate. Students always mix this up, distillate and filtrate. For distillation, it is a distillate. For filtration, it is the filtrate. Okay. Now the liquid collector is known as a distillate. Okay. And one industrial application of simple distillation is water desalination. That means we try to obtain potable water. Potable means drinkable water from seawater. Okay, and that's very important because Singapore don't really have a water source. All right. So I've explained what is a thermometer. Basically, is to check the temperature at which the water vapor is it at 100 degrees. If it's at 100 degrees, it is pure water. How about the boiling chip? The boiling chip is to ensure smooth boiling. Okay, because if there are no boiling chips, there will be bubbles form when the water is boiling. Okay, so I want a smooth boiling, so I put in some boiling chips to cut down on the production of the air bubbles. Okay, the last method is chromatography. Okay, so chromatography is used to separate colored components in a very small sample of the liquid mixture. Okay, so when the components in the mixture dissolve in a solvent, we need something to dissolve it. They will spread out at different speed. Why do they spread out at different speed? Because the more soluble, the further it will spread. They have different, remember we are doing mixtures. We are doing separation of mixtures. And in the mixture, they retain their original properties. Okay, example, like I have A, B, and C. Okay, they are mixed together in this liquid, right? A, maybe it dissolves fastest. Okay, C is slowest. Okay, so they will spread out at different rates. And that is the principle behind chromatography, right? The more soluble the substance is in the solvent, the further it spreads. Okay, so how do we use chromatography? Right, this is the pure dye. We are using it as a control. Okay, so the pure dye will dissolve in the solvent. So all these are solvent. Basically, you will put this, okay, in a solvent, maybe somewhere here. Okay, and the solvent, they will creep up because of the movement of the solvent. So as it goes up, it will actually cause the pure dye to dissolve in it. And the pure dye will form one spot because it's pure, right? Now, if we look at the impure dye, let's erase this. Let's look at the impure dye. The impure dye means that there is more than one component. It is not as pure as the pure dye. Okay, so it actually forms 1, 2, 3 spots. Okay, so it separates into 3 spots. It's a mixture with 3 components. And you see that 1, 2, 3 don't tally with the pure dye's signature. It doesn't form, it doesn't align. Okay, it doesn't align to the pure dye's spot location. Okay, that means the impure dye doesn't have the dye component in the from the pure dye. Okay, and how about those dye insoluble in solvent? Okay, insoluble, so it will still remain over here, so there won't be any spots. Okay, so how about now, let's say there is in the impure dye, there is also a signature here which aligns perfectly with the pure dye, which acts as a control. So we know that, oh, actually the impure dye has, other than the pure dye's component, which is this, it also has 3 other spots. 1, 2, 3. So then we have to go and find out what are these 1, 2, 3. Right? So we can use chromatography again, and if we suspect that maybe it's a certain kind of dye, we can put it here again, and it should show, it should have this signature or this spot which corresponds to the impure dye's chromatogram. Okay, so applications of chromatography are separate the pigments in flowers and leaves. Okay, so because they have different properties, so some will dissolve faster, then we can actually see what are the spots that are created. Then we can tally with a known control. Then we can see, oh, actually what is found in the leaves are from there. Okay, we can test a substance for purity. So example, the impure dye, we know that it's not pure. We can identify components in a colored mixture, detect drugs in urine sample, and detect forgery in checks. So all these are the applications, so it's good that you memorize them. Okay.